Good morning, good afternoon, good night, or evening with everybody. At this moment, we're going to talk about the vegetable histology and how that works. As well as that, what we're going to do is to discover how these structures work. We're going to discover the three different types of structures we have in these cells, as well as the difference between microscope and microscope. So let's begin with it. It's nice to present the group 5, which is integrated by Lisette Zimbania, David Quito, Wilmer Illescas, Brandon Garces, and finally Natalia Marquez. Well, firstly, before we start discussing about these cells, we have to have in mind what is the clear definition of a vegetable histology. Although you might know that histology is the study of tissues, is the study of the microstructures of the tissues of human beings or animals, the same is true with vegetables or any plant that you have. The reason for that is that when you study, for example, a plant, imagine that this is a plant, and you study the properties, the characteristics of this plant microscopically, you're going to find some patterns, you're going to find some figures, some shapes, and in identifying those shapes, we're going to end up discussing and having an idea of them. So, firstly, here you have an image of a histology vegetable plant. Firstly, we can see here that a lot of things are formed, such as cells. One thing to have in mind is that a cell is composed of 3 different structures. The one that is in the central part, which is called xylem. The next one, which is in the middle between, is going to be called cambium. And finally, the last one is going to be called phloem. The main thing to understand about these 3 different properties, these 3 different characteristics of the cell, is that all of them are important. One thing that we have to have in mind is that these cells that are inside, the ones that are called xylem, they only can flow in just one direction. They just can only go, for example, from down to up. On the other hand, the ones that are the phloem, they can go up to down, down to up. They can go to any different point. And the reason is clear. Here you have an example, other image about what I'm saying. The xylem is in the center part, the cambium, which is the one that is between them, is in the middle, and finally the phloem, which is in the last part of it. Well, to discuss it properly, we have to know that the phloem is like the stem of a plant. Imagine that this is the stem of a plant. The phloem is going to be in the opposite sides, in these 2 parts, whereas the xylem is going to be in the center part. Therefore, the phloem can go from up to down, down to up, because when they're going to absorb the nutrients from the floor, from the soil, they have to do it. Like, I'm going to absorb the nutrients from the floor, and then I'm gonna carry them to my other parts of my stem. And well, so what is the cambium about? Well, the cambium is actually this part that is gonna help me to create something which is called grafting. Grafting is this thing that we do a lot in Ecuador. It's a method to reproduce some plants. Imagine that here I have a plant that has been growing for a lot of time. And it has a lot of nutrients, has a lot of vitamins, and all of these things that make them a strong plant. And here I have a stem of a plant. It's like a small. So this stem of a plant, we're going to put with the cambium, we're not going to connect them. And this is going to grow quickly. Well, then we have 2 things. We have to know that we went to the laboratory, and then we had 2 important things that we learned to use. The first one is the microscope, and the second one is the microscope. Well, here between the microscope and the microscope, we're going to see a difference. To identify the difference here, I present 2 examples. Here I have what is called a microscope, and on my right, what is called a microscope. By the way, I'm there, of course, like We were in the laboratory. And, well, what is the difference between them? It's easy. First, microscope, something that you can see, something that you can actually see, but it helps you to identify it better. Imagine that I have here a stem of a plant and I want to see the dots of it. So I use the microscope in order to see the dots better. On the other hand, microscopes are used to see more than dots or things. We want to see more to see the cells, like the xylem. So that's how they are used. Well, so that's the only difference between them. And these two elements, these two tools, were the ones that we used in the laboratory 2 weeks ago. Well, so here I present two examples of everything that we were discussing about. In this case, we have the first and the second image. Firstly, here, what I present is a plant which is called stolons. Stolons are actually plants that they grow horizontally. The roots are like in a line, and they absorb the nutrients in that way. It's important to mention, of course, that these stolons have something called bud. This bud is like a green part that helps them to identify. Well, for example, here we have the stolon. We have here the bud. If the bud is in the side where it receives the light of the sun, this bud is gonna be creating cells in order to generate like leaves. On the other hand, if this bud is below, underneath, this part is gonna identify that there is not light and there is also like the soil. So for that reason, it is gonna be, the roots gonna be like starting to make them to create roots. So that's how the buds work. And then we wanted to see that in detail over the microscope. So here you can see the microscope on your right. So here what we identified is like these three properties of the cell: the xylem, the cambium, and the flow. So we identify here, and we can see here the small cells, everything of those small cells, and we were able to see that. You know that since we are using here a microscope, we were able to see them. On the other hand, if we had used, for example, the microscope, we would have never seen these structures because it's not possible. You remember that microscope can only be used in order to see things that you can see with your eyes, but in a better way. So that would be everything that we learned 2 weeks ago in that Productive Agile Laboratory part. Well, and that will be all that we learned today. We learned today about cell structures. We learned about what is the cambium flow, and finally about microscopes versus microscopes. I really hope that you appreciate this video, and well, the final word will be thank you. Thank you so much. This was a group 5, and for more videos than for this, like this, just follow us. Thank you and bye-bye.